So, hello everyone. I'm here representing my group at Roche. We are working in investigative and immunosafety in the preclinical area. And I'm here to talk to you about how we're using basically the systems that we've been exploring and seeing in this conference, and how do we use them to do decision making, basically. So what are our challenges and where we're going? And I'm really excited because all that we've been talking in this week is steps that we're all together doing, climbing a ladder to change the status quo. And this has happened before. So I want to tell you a quick story about how you change the status quo. So a long time ago, people thought the Earth was the center of the universe. But some of the data wasn't really fitting. So the planets were doing these weird motions, and they try to make sense of it and make models and theories about it. And it didn't really fit. And so some people came with innovative ideas. And one of them was Nicholas Copernicus, who said, how about we change the focus? Let's put in the center, the sun. And it's just really elegant, you know? But of course, nobody believed it because there was data missing. So it took actually centuries to collect this data. And one of the key players was Tycho Brahe, who collected through the whole of his life with very advanced instruments for the time, the movements of the planets. Now, this data could have died with him because he wasn't really good at sharing. But luckily, one of his students or collaborators in that time, Kepler, knew where to find this data, and he could then take that data after Tycho Brahe died and come up with new ways of modeling the data to prove that innovative idea that Nicholas Copernicus had before. And then after him, when he said, okay, actually the theory works if you just make it like an ellipse, and if you this change in mindset, and then came Galileo and Newton, and the rest is history. So actually, it's very important, that step in the middle of when we collect that data, because that is what allows then the new ideas to become accepted. So where are we today in our climbing of this stair? We need to do better, basically. So our translation rate is still quite poor. There's a very low likelihood of a drug being approved in phase 1 to actually go through the whole process and get final approval and be there really for patients. So we clearly have to do better. And we already have some ideas. And it's a matter of saying, okay, what else do we need to make our case and to really come up with these better models to make those predictions? So I think here is a community that we are aware of these challenges. So what is making it challenging? One of the topics is obviously humans are not animals, and that's why we are all here working on it to make more human relevant models, aside of also the ethical issues of reducing and even replacing animal models. So this is not new to this community, and it's one of the challenges that we are trying to overcome. What are the other things that are making our life challenging? Is biological diversity. So why do we respond different to treatment? And we've heard a lot in the morning and throughout the whole week, personalized medicine, personalized making predictions. And still a lot of our programs rely on cohorts of patients where we have non-responders, we have people who respond okay, and people who respond really well. And what we need to start thinking is what do we learn out of these exercises, and what do we learn from what happens in these rounds of testing, so that we can bring those learnings back and start with forward translation. So this is another one of our challenges. Now, it is also very difficult, especially for us in safety, because we have a growing arsenal of therapeutic modalities. So, and this is just one example. There's many therapeutic modalities, but by specific antibodies. And just to make the point, you can see that there's a plethora of molecules, and they all have different biology that we need to replicate in an in vitro system to be able to actually achieve better predictions and be more close to what these antibodies do in vivo or in patients, basically. I was very happy to hear already that we're already aware of reproducibility and trying to change that. So we see that also even, for example, this is one of my favorite initiatives, a reproducibility project for cancer biology, where they assessed like, okay, from all these top tier papers in cancer research, which ones can we reproduce? And it's been a huge effort to do this. And it's quite disappointing, actually. So the reproducibility is very low. But what I like about this consortia is that they're not just focusing on pointing out the pain point, but they also are picking up the key learnings. So they are pulling what makes these papers that you see in blue here, what makes those papers reproducible. And it actually boils down to reporting standards, good documentation, very defined and detailed recording of experiments, materials, data, code, how things were analyzed, and then that this data is registered, that there is transparency, that you can go actually to the people who did it and have that access to that. And then also that there are these incentives for replication, which is, I'm very glad that this discussion was already happening. So these are also, it's not just pointing out the problem, but also like, let's look at the solutions and go through them. And especially for us, this is incredibly important because the learnings on translational power arrive years after clinical trials start. So if you see here, we have like a couple of drugs that have been already approved, and you can see in the colors are like when the clinical trial starts, so phase 1, until when it's approved. And you can see that it takes a long time. And on the bottom, I highlighted that. So it's really decades. And the learnings, so you can see the publications here, they come even after that. And still, we are in the gray zone there in the beginning where we have to make decisions today. And this is where we are getting stuck. So nevertheless, we do put molecules in the clinic, and actually we do it without animal studies. This is a package where we enabled an entry into human in vitro only safety regulatory package. And this was actually a golden opportunity for us because it was a molecule, it's like the TCR-like molecule that binds to a peptide instead of a protein. And this molecule is working in the context of the MHC complex. So it's very human specific. And because of this, animal safety testing was just not possible because of the mismatches in the human-animal MHC and also because of the peptidome differences. So here we have a huge challenge and at the same time, a huge opportunity to show that we can actually put a molecule in the clinic without animal models. So what you see here is, what we did is take all the critical organs and cell lines, and not cell lines, sorry, cell types for that we would need to screen to really come to like a comprehensive package to show we're doing the best that is in our hands to give an in vitro only package to the regulatory authorities. And what is interesting is that this is a huge endeavor. So we're including 2D cultures, organoids, spheroids, macrophysiological system, and we are working internally with our labs and our own internal development groups. At the same time, we also work with collaborators. We also work with CROs, so external providers. And this is a mammoth task to put together these packages. And obviously, related to that, and because we have such a plethora of systems involved, we have very variable readouts. So we do cytokines, we do PCR, we do functional low-dimensional readouts, we do microscopy, flow cytometry, omics. And you can imagine that if for each one of these, we're creating all this data, it gets big, basically. So, but let me go back to, okay, how do we even like start to choose which systems to use for this kind of packages? And we do this in very tight combination with what project needs, so what the molecule needs. So we look at physiology, that's obvious. We see like, okay, do we have like a good level of physiological relevance in the system? That's one thing, but we also look at target expression. So which model is basically reflecting the human physiological situation in the different parts of the organ. And based on that, we can compare among the systems that we have and make the best choice of the system that will help us answer those questions for each one of the organs. And then obviously there's also technical questions. So regarding like accessibility of the tissue and so on. I will not go into it, but it's not just that part, but we also need to basically put the thing together in physically the plane. So, so the other thing that we do is we have to do the model characterization embedded in the safety assessment. So basically we cannot wait 4 or 5 years until the model is characterized because we have to put the package together today. So what we do is we embed this characterization within the safety assessment. And actually this is like crucial because a lot of our data then, if we see noise, if we see something that it doesn't really make sense, then we help ourselves with that characterization to define whether this is something because we are working with the heterologous co-cultures or whether this is something related to the PBMCs that we used. So we had some fantastic posters in the session from my colleagues that are really pushing all of these initiatives. If you didn't have a chance to see the posters, I invite you to revise their abstracts and contact them directly so they can give you more information on this. And we do things like ranking our PBMCs, HLA matching, the heterologous co-cultures, and also revise the physiological relevance of the models that we're using. So no, it's probably not perfect. We are heavily working on it, but we need to do it also in parallel with the actual drug development. And actually by doing that is how we learn what we're still missing to know about the models. So you can imagine then we go ahead and we deploy all these models and we put our molecules in. And for each model, for each replicate, for each time point, for each readout, we generate a lot of data. So this is an example only for like kidney, renal proximal tubule, epithelial cells. And you can see that we have all these different readouts. And this is just for one time point. So this is not yet aggregated, integrated, interpreted data, okay? We do this for all of that battery. And so the challenge then is just put in numbers. We take like all these different variables. We end up with literally millions of conditions. So we have multi-model, multi-readout, complex integration, complex interpretation even. And somehow we need to make sense of this so that we can say, take a decision basically. And that's not all. We, on top of that, have the whole part of the metadata, which is not included in this number, about like which individuals we considered in the platform, the samples, the experiments, biospecimens. So all of these things are variables actually, and somehow we need to make sense of it, and it's extremely complicated. And now with this level of complication, and what I showed you before about the cycle of the data, which is sometimes even in decades terms, try to think about finding and understanding this data in 10 years' time. Think about your own experiments, like, could you make sense of it in 10 years' time? I would find it a challenge. So what we are resorting to be able to make sense of it in 10 years' time is to implement good scientific data management. And this relies on prospective fair data so that the data that we collect is fit for purpose, already thinking of those 10 years from day one. And that's how you make then this data findable, accessible, interoperable, and most importantly, reusable, so that then we can start going in these areas of knowledge, insights, and for us, very importantly, decision-making. And in practical terms, what it means is we need to record what we're doing. So from day one, from the moment we're planning, from when we're sourcing our material, when we start implementing our experiments and generating the data, we need to know what we did and have identifiers so that we can trace back our results to specific individuals or specific variables that then we can say, okay, here is what is giving us predictability or not. And the other part of the coin is, once we generate all this massive amount of information, we need to synthesize it. So we need to process it, integrate it, and do modeling to then be able to get to this decision making. So go, no go, maybe we develop a de-risking strategy, maybe we prepare a regulatory filing. And really it's about ensuring the connectivity of those readouts so that when the molecules get to the clinic and we start producing also clinical data, that these two data sets match and can be connected. And that is the only way we can then start to increase confidence in our translation power. And we have internally also a huge initiative to verify, so to make this data fair in clinical trials, so that this is enhanced data and insight sharing program. And really the key is that in the center is data management, data integration, and having always a single point of truth when we can refer to where the data was generated. And this is what then allows this forward translation. And also when we go to the clinical setting to really bring that back and achieve that reverse translation. And we have guidelines and practices already implemented for clinical data. So we have our FAIR data playbook. We have guidelines on internal reuse of patient-level clinical trial data. We also have a framework for responsible access to internal data. We shouldn't forget that we are dealing with patient data, which is very sensitive data, and we need to act with it responsibly and have also the proper controls on the access to that data. And nevertheless, we need to be very careful with our data through all this process of planning, acquisition, processing, and release. And so what we're trying to do is, even if there's parts of our work where we are working in parallel within clinical data, that we both escalate this ladder together. So we have a layer of infrastructure. We have APIs, so programmatic access is incredibly important because this kind of complex integration can only be done programmatically. And then we have our scientific data assets, which are specific to the domains where we're working, whether it be clinical, whether it be animal studies, in vitro data. That's very specific to the domain. But then we need to add on top of it a layer of harmonized data that makes the bridge to other domains. And on top of that, we can create an integrated data sets, data marts, that we can then, on top of it, do analytics and have visualization tools that help us understand what is happening. And that's how we get then the transitional power. So it might sound even a little bit dry, the whole data, take care of our data thing. But actually, if we really want to achieve translational power, we need to climb this ladder together. So with that, I would like to thank all the fantastic colleagues. So this doesn't happen if we don't have the support and we don't have a lot of people and a huge community of colleagues really implementing and putting this data together and doing this amazing work, whether it be on the bench, on the analytics, on the IT side. It's really, we're all in this boat together. And I just want to thank all of my colleagues that we climbed this ladder together. Thank you.